Today we'll talk about trees, their biology and associates. I'm Joe DeCola, Director of Research and Development for Arborjet. Okay, next slide. Okay, hang on. All right, I'm having a little technical difficulty. All right, so today what we're going to talk about is just what is a tree, tree biology, obviously, but also I'm going to break that down into tree anatomy and physiology, structure and function, and aside from the tree, some of the associates that trees are related to that help their survival. And then some, if we get that far, I hope to complete this session in an hour, we'll look at some patterns of dye ascent in injected trees. So some of the perspective that we're going to address today is how do trees function, and I will interject in places where it makes sense with respect to tree injection. So there are several definitions of what a tree is. Trees are large, woody perennials with a single stem. Sometimes they have more than a single stem. That's an ISA definition. They are long-lived compartmentalizing perennials. That's an Al Shigo definition. So what is tree biology? Tree biology is the study of structure and function, or anatomy and physiology. Anatomy and morphology is structure and form. Those are the component parts of trees. And physiology is the study of the biological processes, the life processes that go on within trees and plants in general. Phenology is the study of the periodic life cycle of plants influenced by climate. So the basic structure of trees, like all living organisms, basically they're made of cells, and those cells are organized in tissues. Unlike animals, the cells are membranes are bound by a cell wall, which is comprised of cellulose and lignins. Those are further organized into plant organs: leaves, stems, roots, flowers, and fruit. So the anatomy of leaf is, there are many layers. If you were to take a cross-section through a leaf, on the left you'll see an angiosperm or a deciduous tree leaf. On the right would be a needle from a conifer. There is a cuticle, which is usually a waxy layer. It's for protection to reduce water loss. An epidermis. And then there are parenchyma cells as a palisade and a spongy layer. Within those layers buried are the vascular tissues, xylem and phloem, and underneath on the undersides are the stomates bounded by guard cells. And we're going to talk about some of this in detail today. So the stomates are very important because they regulate moisture loss and gas exchange in the plants. They are responsive to diurnal changes, which means that the plant has a circadian rhythm. In general, they are open during the day and they close at night. That's not absolutely true. Some desert plants work the other way. Temperature changes. When it gets very hot in the summer, they'll tend to close, even during the daytime. And they are subject to water vapor pressure gradients, which is very important, as we'll see in a little bit. So what are the functioning, how do they function? How do they really work? So if you imagine guard cells as these two balloon-like structures that are affixed at the apical ends, the top and bottom. So just imagine these two elongate balloons connected top and bottom. If they deflate, if you will, they're closed. When they inflate, when they become turgid, they'll open. So how does that occur? So in light, there is an active pump, hydrogen pump, that is very responsive to light. What happens is hydrogen ions are pumped out, and in response, potassium and chloride ions are move across passively through the membrane into the guard cell. That causes an increase in osmotic pressure, water flows in, and the guard cells become turgid, and because they only can move laterally, the pore opens up. When that pump stops, the opposite happens, and they deflate, and you get stomatal closure. So that we'll see in a little bit is really important. That opening and closing of the stomates is also critical to photosynthesis. And photosynthesis occurs in chloroplasts within the cell. And the equation that we've learned, all of us learned in high school, is carbon dioxide plus water in the presence of light yields a carbohydrate, a sugar, and as a waste product evolves oxygen. So photosynthesis is efficient at 1500 lumens and most efficient between 82 and 95 degrees Fahrenheit. The transpiration and photosynthesis, as I implied, are related, and they're interrelated. So when the stomates are open, there is a loss of water vapor, but at the same time, there is the opportunity to absorb carbon dioxide, and carbon dioxide is critical for photosynthesis to occur. Water is also critical. Both are obtained through the atmosphere. So photosynthesis proceeds in a certain wavelength. So visible light occurs between 380 and 750 angstroms. That's between the violet and red range. It's the range that we could see. It's called visible light. But not all parts of that spectrum are useful for photosynthesis. Green, for instance, is reflected and is a part of the wavelength that is not utilized. Two areas are utilized. Light in the blue and red ranges are used in photosynthesis, and the graph up in the upper right shows that the differences between the spectra of light between a couple of types of chlorophyll. There's A, B, and C. We're mostly going to talk about chlorophyll A and B that are utilized in plants. Some of the other factors or micro elements or micronutrients are required in photosynthesis as cofactors or parts of coenzymes. Manganese and iron are particularly important. They're important for splitting water, for photosynthesis to proceed. And when they are deficient or not readily available, then that's when you begin to see symptoms of chlorosis or yellowing in plants. So when pH of soils are high, for example, in Midwestern states, you may see this occur in things like oak trees. There is a certain relationship that between photosynthesis and respiration. Respiration can be represented in the opposite formula, where you start with sugars, C6H12O6, utilize oxygen, and in that release energy, carbon dioxide, and water. So plants do respire. Roots respire. Roots do not photosynthesize. So non-photosynthetic parts of the plant respire to release energy, but also photosynthetic parts of the plant also respire when light does not happen, like at night, for instance. But there is this relationship between photosynthesis and respiration. And on balance, photosynthesis has to be greater than respiration for the plant to live. So plants are autotrophic or self-feeding. They have to make enough during the growing season, enough sugars for the 5 basic functions, and that's metabolism, growth, reproduction, self-defense, and storage. And storage is kind of interesting because storage, generally starches and oils, but starches that are in storage are utilized in the spring for bud break. So that's the battery, if you will, to start the next cycle of growth. Okay, so there are 2 types of meristems. There's primary meristems that occur at the tips of shoots and roots, and they increase the plant growth by elongation, and the secondary or lateral meristem increases plant size in girth or diameter. In trees, we can talk about 2 secondary meristems. There's a lateral cambium, which produces new xylem or sapwood to the interior and new phloem to the exterior. And within the bark, there is a cork cambium or phylogen that produces new bark or periderm. So there are 4 basic functions of xylem or sapwood: the conduction of water and dissolved solutes, support carbohydrate storage, and defense against spread of disease or decay. Gymnosperms, conifers, coniferous trees rely solely on tracheids for water transport. Tracheids tend to be smaller diameter than vessels, which occur in angiosperms. So flowering trees or deciduous trees will primarily rely on vessels for transport, but they also contain tracheids as well, whereas conifers rely solely on tracheids. There were a couple of researchers in 1839, one was French, the other was German, who independently came to the same formula of describing the uptake and movement of water in vessels and tracheids. And basically, the rate of movement through a plant is on the order of to the 4th power of the radius. So in other words, the larger the radial width of a vessel, the easier or the more rapid or the higher rate of uptake within the tree. How does that really affect tree injection? Well, you'll find that if you're injecting a tree like a red oak, which has many large vessels, it tends to be quicker. Things like elm, American elm, present a very low barrier to injection, whereas hemlocks and pines, which rely solely on tracheids, it's a little more difficult to inject, takes a little more time to inject. So we're going to look a little bit at different kinds of anatomy. We can describe 3 ring porous, diffuse porous species, which tend to be the flowering trees or deciduous trees, and coniferous trees, which are not described as non-porous because they do not contain vessels. So when you see porous, it means there's vessels that are part of that anatomy. So this is under 20x magnification. These were cross sections that I took a couple of years ago. This happens to be black oak. On the left, you'll see about 3 years of growth. The springwood are the large diameter vessels. It's very clear. And that's what's laid down early, and then the summerwood, which are smaller vessels, and tracheids, which are more compressed, occur later. So the early wood is actually, so when you count a ring in a ring porous tree, you start with, if you're going from the bark in, you're actually going bark, cambium, summerwood, then springwood. So it just kind of an FYI on how to look at trees. And then there's the yellow arrows that point to a multi-seriate, which means a series of parenchyma cells. The parenchyma are living cells that are non-differentiated within the vascular tissue. Those cells are really important, and what you're seeing are basically, we call them rays. This is viewed as a radial parenchyma. It also occurs axially, which means along with the stem of the tree. To the right, what you're seeing is an image of heartwood. And if you look at the vessels in the right image, you'll see little, what look like amber occlusions or plugging of the vessels. Those are tyloses, and that's the ballooning out of the membrane of the adjacent parenchyma into the lumen or the interior of that vessel. In things like black oak and red oak, that occurs after a trauma where you get this ballooning. In white oak, it occurs after the second year or so of growth naturally. And it's the reason white oak is generally selected, and plants in the white oak group are selected for whiskey and wine barrels because they are tight because of this effect, the tylose effect. It also kind of presents an obstacle to injection. So in trees like white oak, you'd want to target the first couple of years of growth in order to maximize the uptake, but not so much, that's not necessarily true in red or black oak. They're open for injection. So just kind of know your species and know what you might be coming across. So doing anatomy is a good little exercise and understanding why trees present some barriers to uptake. This happens to be a semi-diffuse tree. This happens to be weeping willow. And you can see this idea of diffuse porous hardwood. The larger vessels are interspersed throughout the growing increment, so you can still count rings. They're not as distinct, but and then then it depends if you're a lumper or a splitter, whether you call it diffuse porous, semi-diffuse porous, semi-ring porous, and so on. You'll see that all through the literature. All these cross-sections were done at 20x, and here is a spruce, and you cannot see the pores because there are no pores, but those tracheids are really small, and you could barely make it out where the dye was injected in the tree, and they're just so tiny. So it really does present a barrier to uptake. More so when you'll see the amber-colored areas in this slide. Those are resin ducts, which are living parenchyma cells that get together that manufacture resin, and that is one of the natural responses, defense mechanisms in some conifers. So there are resinous conifers that tend to bleed when you attempt to inject them. You drill a hole in them, they're going to pitch out some sap. Some are worse than others. Some of the work that we've been doing is looking at injecting trees later in the fall when things actually get cold. It reduces the resin flow and presents less of a barrier to injection. So that's one of the things that we've been observing in making some applications, so you may want to make a note of that. That tends to be helpful. But this comparative anatomy will give you some inkling into why some trees are easier to treat than others. So there are two elements. The trees are kind of interesting and fascinating organisms. They are comprised of both living and non-living cells, and technically they're called symplast and apoplast. The living cells generate tissue like the cambium and the phylogen, which is the bark cambium. Phloem as a phloem rays and parenchyma. The phloem rays occur in the bark. Axial and radial parenchyma occur in the sapwood, but they're living tissues. And they collectively form a webwork, both radially, which means in toward the radius or center of the tree, but also actually with the stem of the tree. And that webwork are living cells. But sapwood, or the xylem in general, those xylem and trachea, those elements are functional only after they die. So they are just a pipe or a capillary, if you will, and they constitute what's called the apoplast. The apoplast is non-living, and you'll see there that living and non-living is important for water conduction in the plant. So we'll talk a little bit about absorption and translocation. So water is essential for all living organisms, for biological reactions, especially for photosynthesis. That's why I showed you the formula CO2 plus H2O. It's very critical for photosynthesis to go forward. It comprises, it's important to keep the plant upright, keep it turgid, and water dissolves solutes. It's how plants get minerals, get essential minerals for actually metabolism and growth. Phloem transports photosynthetes. What are photosynthetes? Photosynthetes are the carbohydrates or the sugars produced by the green parts of the plant that contains the chlorophyll. Movement of water in the xylem and photosynthetes in the phloem are examples of axial transport. That means up and down, essentially, or source to sink. We used the way I learned it years ago was xylem up, phloem down. But basically, wherever there's green that manufactures carbohydrate, and wherever there's growth, there's a requirement for carbohydrate. That's source to sink. Radial transport is the horizontal movement of water and nutrients within the tree. So although you have manufacture of photosynthetes in the leaf, it's wherever there's cell division occurring that it becomes a site for growth. You'll need both axial and radial transport. So transport can be active. That means energy is expended, or it can be passive. So it can either be active through the symplast or passive through the apoplast. In ring porous trees, as I applied before, the 2 to 3 most recently formed rings, white oak, black locust, American elm, are active in water transport. So those are the ones that if you want to inject efficiently, those are the ones that you would target. The older increments tend to tylose. Tyloses are important as a defense mechanism, as I implied. American elm, it didn't work very well, but one of the reactions to infection, Dutch elm disease is the vascular wilt that grows through the xylem tissues. Well, one of its first lines of defense was to put out these tyloses to reduce the spread of the fungus through the tree. It didn't work that very well for American elm. In some other species, it works pretty good. Other older increments do tylose, and tyloses are the blue line protrusions that occur from the adjacent parenchyma. Diffuse porous trees tend to plug older xylem. When the older xylem becomes your heartwood, with gums and resins. It doesn't tylose. The ring porous trees tend to tylose. The transport and storage may remain active for 50 or more increments. That's 50 or more years, and maples are an example. And conifers, again, move water and solutes through tracheids. And they have open pits between the tracheids, and they tend to be open for at least 10 to 20 years. Just want to talk a little bit about plants, because there are mechanisms of control within the plant. And these are plant growth substances or plant hormones. Auxins are important in trophic responses. How does the plant know to grow toward the light? Well, it's auxin. How does the plant root know to grow toward down into the ground? Well, that's a geotropic response. It's, again, auxin. Auxin, interestingly, is produced in the shoots, and it aids in root development. And conversely, cytokinins produced in the roots drive shoot development. So there's this relationship between root and shoot. Gibberellins are important in developmental processes. They tend to encourage stem elongation, so internodal, so between nodes. That bit of stem that elongates is driven by a gibberellic acid. If you were to use a plant growth regulator, that plant growth regulator will that acts to reduce growth. Many of them are. They function as plant growth regulators. Actually interfere with gibberellic acid production. But gibberellins are important, and they were first discovered in Japan in rice. And the Japanese term for it is foolish plant disease, because these rice plants infected with a fungus that produced too much gibberellic acid grew to over 6 feet in height, and so they first termed it foolish plant disease. But that was gibberellic acid. It also is important in germination, flowering, and fruit senescence. Interestingly enough, abscisic acid has very little to do with leaf abscission in plants. It's ethylene that drives abscission in plants and is also a fruit ripener. So ethylene is given off by ripening fruit. And I think we've all experienced this, if you store an apple near your bananas, they're going to brown rapidly. Well, that's ethylene being generated by the fruit. Abscisic acid, though, is produced, even though there is a role in abscission, it's basically produced in response to plant stress. Okay, so root anatomy, what are roots for? Well, they're for anchorage, storage, absorption, and conduction. Generally, the larger woody roots are for anchorage and storage, but not absorption. So just think of those larger roots as the highway. The small fibrous non-woody roots is where absorption occurs. So these non-woody roots, which are ephemeral, they're shed by plants annually, by trees, and regrow. This is where the activity takes place in terms of absorption. It's also where infections occur, and such as mycorrhizal infections or even bacterial infections occur on that surface, and it's the non-woody surface. Movement of water is basically across a moisture gradient. So from a high moisture gradient to a low moisture gradient. That is the basic function of how plants will transpire and maintain turgidity. How does that really work? Well, there's 3 basic concepts. Cohesion, adhesion, and transpiration. Cohesion has to do with how the water molecule is put together. And because there's a strong positive and negative end of water, they are the plus end, which is the hydrogen end, is attracted to the oxygen. And so they kind of cohere together. They kind of stick together. And that's very important for this column of water to rise. The other aspect is that adhesion means that it also, these hydrogen bonds also, those plus and negatives, generally I think the walls are negative. That plus also adheres to the wall of the vessel or capillary. So it kind of sticks to the wall, they stick together, and they could rise. Well, what's the driver of rising column is transpiration. So that column is driven by evaporation off from the stomates, so that column, that water rope, will go up the tree and exit through the stomate. And it could do it for an incredible distance, and it accounts for why some of the largest trees in the world, the redwoods, can grow to the heights that they do grow. But these adhesive forces, the cohesive and adhesive forces, can work over 1,500 feet. It's quite impressive. So, but not only is water taken up through the soil, also dissolved ions move as well. And they are accumulated, they're absorbed through the epidermis. They have to cross the endodermis. The endodermis is a suberized layer of cells, and it kind of forms a barrier. It does form a barrier in the root. And to get across that endodermis and into the vascular tissue, that requires the expenditure of energy. So that is actually, endodermis is symplastic tissue, and there's active transport through the root. So water and 13 essential elements are absorbed by roots. So water moves through the epidermis via apoplastic movement, that's passive movement, but across the endodermis, which is suberized, which means that it's corky. Energy is required to get it into the vascular cylinder. What's interesting is that when there are mycorrhizal, whether they're endo or ectomycorrhizal infections, those occur in the epidermal layer and do not cross the vascular cylinder. So they're kind of external, even though they're occupying the tissues and the cells of the root. It's in the epidermis, and it doesn't cross. Generally, does not cross the endodermis. So we talked a little bit about trees, about anatomy and physiology of trees, and trees don't live alone. They have both mycorrhizal and bacterial associates. Some of the ectomycorrhizae and oaks and pines are actually obligate. They rely on, for good growth, ectomycorrhizae. Some years back I did some work with PT, Physiologus tinctorius, and looked at transplant survival in plants, but there are others. I listed 3 here that are ecto. Some trees rely on endomycorrhizal associates like maples, and one of the ones that you'll see a lot in the trade are Callus species. Some of the bacterial species are also important for tree growth and survival. Some of these bacteria, the bacilli or other genera, but Megatherium, for instance, is an important nitrogen fixer. And so some things like alder, for instance, will have nitrogen fixing nodules on them. Legumes certainly do, but some of these species, Amyloliquefaciens, Subtilis, and Megatherium, are they exist in the rhizosphere, which means the soil root interface, and they'll form a biofilm. So these are protective. And they have different modes of activity to exclude other organisms, other pathogens, for instance, whether they're fungal, etc. They could be bacterial, but they work by competition, antibiosis, which means they'll produce proteins that will discourage other things like Rhizoctonia, Pythium, damping off kinds of things. So they are protective. And then there's some neat species like Rhodopseudomonas and Rhodospirillum that are not only nitrogen fixing, but they're photosynthetic. So they're kind of cool. So you have these inorganic ions. I'm just illustrating some of the major ones, the macros that in the forms that they're absorbed in. But essentially, some of the nitrogen fixing, where does that nitrogen come from? It comes from atmospheric nitrogen, which is N2. It's fixed by these bacteria that are nitrogen fixers, and they will excrete either the nitrate form of it, which is NO3, or an ammonium form, which is NH4+. Phosphorus is interesting, and phosphorus is generally available in soils, but not bioavailable. What does that mean? That means that phosphorus exists, but not in a soluble form. It needs to be mined, so it's because of it forms chemically. It'll react with elements in the soil. Fortunately, this is the role of the mycorrhizae. The mycorrhiza will excrete acids to the exterior and solubilize the phosphate and absorb it and provide it for the plant. So that's the basis of, part of the symbiosis, where the fungal associates provides some of the macros to the plant in exchange. Since these mycorrhizal fungi are non-photosynthetic, the plants provide sugars to those associates. That's how that partnership works. So carbon, hydrogen, and oxygen make up carbohydrates, basically sugars, starches, etc. And those are obtained essentially carbon dioxide from the air, water from, it could be from the atmosphere, but generally it's uptake through the roots. And then the 13 essential elements are absorbed via the roots. So 99% of plants are made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. We talk about macronutrients or macro elements as NPK, Cal, Mag, and sometimes sulfur. Micro elements or micronutrients are just as important. Manganese, iron, that I talked a little bit about, being cofactors and important in photosynthetic processes. The difference really, they're all essential, they just are required in different amounts. So difference between macros, which are required in percentages, micros are required more in parts per million. So if you are to approach a tree and you want to fertilize it, fertilize the soil around the tree. But if you have issues with high pH and intraveinal chlorosis, yes, you can inject the micros because it's a smaller quantity and it lends itself to injection. Okay, so I just want to talk a little bit about carbon-nitrogen relationships, and I just picked a molecule, an important one in trees, chlorophyll. And what you can do is take the molecular formula and look at their carbon-nitrogen ratio. Essentially, your carbon to nitrogen ratio is basically anywhere from 9 to 14 parts to 1. What does that really mean? It means that plants are adapted to manufacture the carbohydrate part of it, but they have to absorb the nitrogen part of it, of the element. So as a nitrate form or an ammonium form, and that's where the associates come in. So where they're good at taking carbon dioxide and water and making carbohydrate, which is a precursor to many metabolic processes within the tree. They do need these cofactors, and nitrogen is one of them. There are others. While we're talking about chlorophyll, the central molecule in that porphin compound is magnesium, and that and it may be of interest, where chlorophyll and is a porphyrin. Your hemoglobin is also a porphyrin, but the central element in hemoglobin is iron and not magnesium. Okay, so just to kind of summarize root transport, root from roots to shoot and shoot to roots. So transport within the plant goes in both directions. Roots absorb water and dissolve solutes and basically move it to area from that source to sink, basically apically into the plant leaf. And then there's radial transport that occurs from the outer layers in, and that could be both symplastic and apoplastic movement. And leaves transport photosynthase sugars to where it's needed. Basically, we think about roots, and that goes through the inner bark. So the inner phloem is which moves, and there's different theories on how those sugars are moved, but it's through the inner phloem. Just want to talk a little bit about compartmentalization in trees, because this is part of tree defense. Trees are highly compartmented. We talked about growth rings, which are increments, and we looked at the anatomical feature, and then we looked at how those increments are bounded by parenchyma, and we looked at some cross sections that did that. And the parenchyma is wider in things like oaks, and they were very small. You couldn't see them in conifers. Not that they don't exist, but they're just single cell thickness in conifers. Shago talked a lot about CODIT, which is an acronym for compartmentalization of either discoloration, defect, disease, or decay in trees. Pick the D that you like. There are 3 distinct reaction zones. They may be described as wall 1, 2, and 3, which have a very definite orientation. So wall 1 limits the vertical spread, so that's with the stem, up and down. Wall 2 limits the radial spread, or inward toward the center. And wall 3 resists the lateral spread, or the circumferential spread. And those reaction zones generally are arranged from weakest to strongest, So you think about it, the vertical limit, and I was talking a little bit about Dutch elm disease, not being limited by tyloses, but not so hot. Well, trees have to move water and solutes vertically, so to impede that, it's not in the tree's best interest. But in order to resist girdling, that reaction zone, being wall 3, is very strong. So trees don't heal. Whatever injury occurs is there for history, and you can date that. I'll talk a little bit about that in a bit. But after the wound occurs, the lateral cambium will form a new boundary. And that is, so after wounding, you have these parenchyma cells, you have meristematic tissue there that are not differentiated. They're initials. They can make new tissues, and they begin to divide, and they could form this closure tissue, or wall 4. So that wall 4 separates the wood at the time of wounding from the new wood. So the neat thing about that is you can date when that wound first occurred. So trees and plants in general have a system of defense. Bark, thorns are important for larger animals. Like why would plants have thorns on them? But larger mammals or birds that consume trees, well, they're going to defend themselves. Leaf hairs, thick cuticles for smaller organisms, cellular materials that resist decay for microorganisms, and chemicals that resist insect feeding, pathogen infection, and decay. So there's all these modes. They're both anatomical and physiological for tree protection. So our CODIT walls, are they strong or weak? This is largely under genetic control. So the tree response is under moderate to strong genetic control. So some trees do really well that resist injury and decay, and some trees are lousy at it. And so what does that really mean? Well, trees in wooded areas that are grown from seed will vary in their CODIT responses. Trees that we get from nurseries that are produced clonally by cuttings will tend to act the same way, and you just have to learn your trees. So when a barrier is within the growth increment, it is possible to date the time that wound occurred. This is kind of cool. So I just threw this in here. So most species, the growth increment starts to form about leaf formation, so when leaves start to form, and 90% of that increment will be completed 6 to 8 weeks later. So if you see, if you cut a tree down and that wound response that you're looking at occurred within the middle of the increment, you can calculate that it occurred 7 weeks, approximately 7 weeks after leaf formation normally started that particular year. Okay, so a callus and woundwood, what's the difference? Callus is really undifferentiated tissue. They're just uniform cells, no lignin, and they're meristematic, and they can form whatever they're going to form. Woundwood is already decided what it's going to form. So it's either going to, woundwood will form new xylem tissue to the inside and phloem tissue to the outside. And you can see that occur when and after a tree is injected or wounded, even by pruning. So there are also natural infective agents that attack trees. Some are introduced, like Dutch elm disease, Ophiostoma nova ulmi, was one of the earliest. It's introduced around in the early 1900s, and kind of swept through and killed many of the American elms. The American elms were planted on Main Street, USA, part of the centennial celebration, and they were produced clonally, so when a disease came through, like Dutch elm disease, it kind of wiped them out. Oak wilt, another phyto-pathogen that we happen to be interested in and working on. Sycamore anthracnose, one of the new diseases down in Florida, Texas phoenix palm decline, Phytoplasma palmae, bacterial leaf scorch, and fire blight, Orinia. These are all maybe termed phytopathogens, but they occur within the plant tissues. These are the bad guys in general. There are some good guys, too, that may be associated with tree tissues that occur within the sapwood of trees. Some occur naturally in soil, like Trichoderma. There's some Bacillus species, Enterobacter, and Pseudomonas species. Enterobacter is probably the one that we're most kind of acquainted with because it, some Enterobacter species actually form a bacterial wetwood in trees, and that bacterial wetwood is competitive with some fungal decay organisms and form a kind of protection wood within the tree and prevents decay within the tree because it actually modifies the environment within that sapwood tissue that it occupies. So they are potentially antagonistic to those phytopathogens I talked about by allelopathy, ISR, antibiosis, competition, or parasitism. So what does that mean? Allelopathy means that the organism can excrete other chemicals that suppress the competitor. ISR is induced systemic resistance, and what it does is it actually incites the plant to defend itself. And it does that through a pathway called jasmonic acid pathway. Some people, you may be familiar with systemic acquired resistance, SAR, and that's a different pathway. That's a salicylic acid pathway, but the results are the same. It's basically taking sugars that are in storage for defense and turning them into antimicrobial compounds to defend the plant. And some of these endophytes do that. And so that becomes an interesting line of research. So I just wanted to kind of wrap up here with some illustrations, some sections I did years ago when I first looked at injecting trees and did some dye work. And what was kind of interesting to me was you can, it's kind of how we designed injection into trees, but if you look at where the pink dye occurs in this red oak, it occurs in the sapwood. Sapwood is lighter color, and heartwood is darker color here. And actually, what I did was drilled into the heartwood, injected the dye, but you can see where the dye, and then cut the tree section, and you can see where the dye kind of perfused. So there are tyloses and extractives that are deposited within the heartwood, so it's pretty inactive. So water, and in this case dye, will flow through the least resistance, and you can see it's throughout the active increment. And this happens to be red oak. So that was kind of interesting. If you look at it from a axial perspective, where we split the wood right through the injection site, you can see where you put the infusion legs. You'll see the dye movement. And dye movement will occur both upward and downward. The net movement, though, is upward in trees that are actively transpiring. In white oak, you'll see in this particular specimen, it's one increment was open, and the others looked tylosed. Generally, white oak has at least 2 or 3 open for transport. So white oaks can present more of a barrier to tree injection, as I applied earlier. But you can see that just because some of those elements are tylose doesn't mean you absolutely do not get movement through those increments. And here we see movement of dye through the 6th increment. In willows, the movement of dye is strongly axial, up and down. No big secrets there. This happens to be a semi-diffuse forest tree. And some of the sections that I did in dogwood, for instance, was to, and I do this a lot, is to section the tree from the base at the point at which we inject it right up through the major scaffolding branches. And you do this to kind of look at the pattern of movement, because all these different species move dye and injected products differently. So essentially, when you inject into the stem, the ascent up the tree tends to be within a sector, which means in an area. And it's why we recommend in trees to go to multiple injection sites, because it stays within one area of the tree. So it's a fairly narrow column, but as you go up the tree, that column actually gets wider as the tissues compress. And it almost completely surrounds branches, because those are the most compressed tissues. So you get this lateral movement. As you go upward, the spread and coalescence of the dye increases. And then you can see in the picture on the lower right, the dye actually occurred pretty much completely within the smallest twigs that we dissected. So we got pretty complete dye movement, and it was part of the research that we did early on to determine the number of injection sites around the tree. In conifers, and this happens to be white pine, you'll see a dye going up through the column. You could also see some this happens to be a resinous conifer. This is a branch that got dissected, and although you can't perceive the tracheate elements, you'll certainly see movement throughout the plant as well. Hemlock is a little different. It's non-resinous. I mean, the resin ducts occur in the bark and not in the wood. But in conifers, they're a little bit different than the hardwoods. The injected movement of dye tends to be in a clockwise spiral around the tree. And there's some others, some of the research that was done by Kozlowski looked at larches and spruces and documented some of that. And what we found was that in conifers, when conditions are conifers, because they rely on tracheas, they tend to be a little tougher, not only because of the diameter of the tracheas being so small. I liken to describe injecting through a capillary rather than a vessel. But however, because of the limited moisture movement in conifers, you just have to adjust the time of day or the seasonality. I implied earlier that late fall is a really good time, and it's because you're not getting the temperature extremes, which will shut down those stomates fairly quickly in conifers. If you're down in Florida, for instance, I've done some work on slash pine, and I had to get out and try to make some applications. This is some early work I did with triage, and it was like between 6 and 9 in the morning during the summertime, because otherwise it just shut down. And that's not necessarily true in oaks, even in white oak. You can, because of the size of those vessels, you had the entire morning up to almost 2 o'clock in the afternoon. So they're very different. So again, just know your species. And I think that's all I got. So are there any questions for me?